Okay, hi everyone. This is Mrs. Mirando talking about antibiotic resistance and how it's an application of Darwin's theory of natural selection in combination with what Darwin didn't know about, which is genes and mutations in genes. So I'm going to do a quick recap of Darwin's theory ideas like we talked about last week. Let me get back up to the beginning, excuse me. So just a quick recap of his main ideas. We used an example of a giraffe and tried to explain how Darwin's theory applies to how giraffes are so tall today and have such long necks today. So his theory would say that giraffes must have had many offspring a long time ago, and that is a concept called overproduction. And so maybe it was an environment where different length necks could all survive. Maybe there was plentiful food up at the top of the tree and then down at the bottom, and all different length necks giraffes could survive. So there's variations in lengths of necks. Some are tall with long necks, some are short with shorter necks. Variations, variety. But at some point, they probably had to compete for a limited amount of resources. That's the idea of competition. And if you're competing for limited resources, not everyone is going to be able to survive. So in this situation, having a long neck is a survival advantage because you can eat food high up on a tree that the short neck giraffes can't reach. So short necks ended up being a survival disadvantage, and so fewer of them are able to survive that environment with a limited food supply. This concept is called survival of the fittest. So it's not about strength, it's about who's the most fit for their environment, meaning best suited for their environment. And then in the end, if long neck giraffes were more likely to get food, then they're more likely to survive long enough to reproduce and pass on their trait. And the long neck trait perpetuated throughout the species, and eventually the population evolved to only have long necks. So that's Darwin's explanation of why giraffes are tall. So this is a summary of his key ideas, and we're going to apply these key ideas to seeing how and why antibiotic resistance and pesticide resistance has developed in our modern world. So overproduction, variation, competition, survival of the fittest, reproduction, and possible speciation. How do these apply to the evolution of some species like bacteria and pests like insects that are becoming resistant to the drugs that we use to try to kill them or the chemicals that we use to try to kill them? So how does that happen? This is an excellent video, and I believe I made my settings so that you can hear the sound on my computer. So, but I also am going to put on the closed caption.
My name is John Chambers. I traveled deep into the Amazon jungle with my team where I uncovered.
Okay. So this is a reliable source. This was put together by Harvard Medical School, and they're going to explain this little experiment. It's a quick little 2-minute experiment. It's really fascinating to watch. It gives you a good visual to understand how bacteria are able to develop resistance to the drugs that are meant to kill them that we call antibiotics. I'll let him explain.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that Bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
So that, I think, is a big eye-opening and scary thing to see, that in only 11 days, we can see how quickly bacteria are able to evolve thanks to random genetic mutations. That is the key thing that some of you were missing out on answering the ties questions from last week that I just rated yesterday. So that's what I wanted to drive home. That's the one thing Darwin didn't know about. So when we take Darwin's idea of natural selection, these key main ideas, and we add in what he didn't know, he did not know about genetic mutation, because there was no discovery of the gene yet. There was no discovery of, DNA and nucleotide sequences. He didn't know about that. So his biggest weakness was that he could not explain the genetic basis for all those variations he saw. But now we know those are genetic mutations. This is just a quote from his book, him trying to say how he that the mutations in life was breathed in maybe by some higher being, which he didn't know about mutations. So what do we know about mutations now? We know that it's just a change in DNA. It's a random change. It's just spontaneous, and you cannot stop it from happening. So those mutations you saw in the bacterial plate, where they moved into the stronger concentrations of antibiotic, were random. Those bacteria just got a lucky random mutation that led them to survive the higher strength antibiotics. This is how variation exists within a species. And this is why we have variety of eye color, variety of hair color, variety of tolerance to diseases. Some people are able to battle and combat a virus more easily than others, just thanks to their genetics that affects their immune system. So we see when you have a trait that helps you survive in a species, the species overall is going to see an increase in that trait in its population because it helps aid survival. It's favorable. Same thing happens with unfavorable traits. We see those decrease in a population. They're not completely eliminated though, because random mutations still exist. Recessive mutations exist, so you can be a carrier for a mutation that's unfavorable, and then it can pass on to your offspring. So we don't see them completely eliminated, but we definitely see favorable traits increase in number compared to unfavorable traits decreasing in number. And then bottom line is, it still comes down to the environment. The environment is going to be the determining factor that says, well, that trait you have is going to work out well here, or that trait that you have is not going to work out well here. And that was one of Darwin's main underlying theories and ideas. That's why he called it natural selection. He said that the environment, nature, selects for who will survive. And if that environment changes, then something that was once favorable might then be unfavorable. And if something that was once unfavorable could end up becoming favorable in a new environment. So it's up to, or determined by, the environment who will survive. Okay. Also, the more mutations that exist within a species population, the more chance they will have for evolution. The more chance they will have for surviving changes in their environment. I thought this was a super interesting story that I came across, I think, last year in the news. So if you don't know, that African elephants are poached for their tusks. Meaning people go in and illegally hunt them and kill them and cut off their tusks, and then they sell their tusks for ivory, and it's used to make jewelry and trinkets. It's like a status symbol to have something made from ivory. But it's unethical and it's illegal, and elephants are protected. And what we've actually seen happening is an elephant that has a mutation or has a trait where it doesn't grow tusks, it's actually a survival advantage in this poaching world. So we're seeing more and more elephants being born tuskless because the tusked elephants are getting targeted and getting killed. So they're not surviving to reproduce as much as the tuskless elephants are surviving to reproduce. So those key concepts, survival of the fittest, are being applied in this changing environment. Now, it's humans that are changing the environment in this case, because we're creating poaching and creating a market for poaching. And so we see a change in the species traits because of the change in the environment. More and more elephants are being born without tusks. I think there's some tuskless elephants for you. I like this picture. It makes me think of the scene in Soarin' in Disney World at Epcot. I miss my Disney. So how are we doing this? We are having an impact on natural selection. We as the human race, we are having an impact on environments that are going to affect natural selection. Let's tie in. Okay. So pesticide resistance is one example of what we're doing. So pesticides are chemicals we create like Roundup and other chemicals that we create to try to kill weeds, to try to kill rodents, to try to kill insects. Pesticide is just a chemical that's meant to kill. That's what the side means. It means kill. So we're trying to kill pests. We use these chemicals over and over again. Eventually, they don't work anymore. That's going to be a big problem because then we have to create a new chemical that's possibly stronger and maybe has more other impacts on our environment. So how come these pests are becoming resistant? The reason, the cause of the resistance is just plain old mutation. Random, uncontrollable, spontaneous, lucky mutation if you happen to be a pest. Some insects just end up with a random mutation that makes them naturally resistant to the chemical pesticide that we use. So they survive and they reproduce. So we spray our pesticide and we kill all the typical non-resistant pests. Who's left? Only the resistant ones. And when there's only resistant ones left, they have no competition, one of Darwin's main ideas, and they're therefore the best fit to survive, another one of Darwin's main ideas, and we see them reproduce, another one of Darwin's main ideas. And so as a result, that random mutation gave them a lucky survival advantage, and we see an increase in resistant populations. So then we end up saying, oh no, now this chemical doesn't work. We've got to come up with a new chemical that will kill these insects. And so we make stronger and stronger chemicals, and it's kind of like we're on this treadmill of trying to keep up with resistance that is developing by just random mutation, that those pests are just lucky that they end up getting and that they are resistant to our chemicals. The same exact concept is happening in bacteria that are becoming resistant to our antibiotic drugs. Antibiotics are medicines we've devised to be able to fight bacteria. Remember, they don't kill viruses, they kill only bacteria. And they work by sometimes stopping bacteria from reproducing. Sometimes it can destroy the cell walls, the membranes of the bacteria. But however they work is indifferent at this point because some random bacteria just end up with random mutations that cause them to be resistant, naturally resistant. It's a random, spontaneous change that they happen to get lucky and have. And then we use our antibiotics and kill only the non-resistant bacteria. And so as a result, the resistant ones are left with no competition. They're going to be the best fit to survive in their new environment with antibiotics being sprayed around, and they're going to reproduce, and we're going to see more bacteria that are resistant to our antibiotics. Penicillin was our first antibiotic that we discovered back in the 1950s. Penicillin doesn't work on very much anymore. Penicillin is still prescribed here and there for some things. I know some of you are probably allergic to penicillin. That's a different story. But it doesn't work as well as it did when we first discovered it. Why? Evolution. Natural selection, random mutation in bacteria, and overuse of the drug. The more we use it, the more we provide an environment for the resistant, that are naturally resistant bacteria to survive. We're giving them an environment that they're best fit for when we use these antibiotics. Now, does that mean we shouldn't use antibiotics? Of course not. They have helped treat millions of people and save millions of lives all around the world. But we have to be very careful and selective when we use them, how much we use, and how to use them. And of course, there's a different variety of antibiotics with different strengths. So if we overuse either of these substances, our chemical pesticides or our antibiotics, it can result in their eventual failure to work. And then we've got to work on developing new ones. That's a big takeaway to realize. So why is this happening so much in insects and bacteria? Well, this boils down to speed of reproduction and who's more likely and who's less likely to evolve. So if you reproduce sexually, you have more genetic variation, you have more mutations, and you have a shorter, faster reproductive rate, like a shorter life cycle. So you have a faster reproductive rate, like a fly only lives for like 2 weeks. So it's got a faster reproductive rate, so they can evolve faster just because their generations are closer together than, let's say, human generations. We're typically an average of like 25 years to the next generation. You can say bacteria, the next generation is 25 minutes away because it doesn't take them long to split in half. Who's less likely to evolve are going to be those who reproduce asexually and have less genetic variation and therefore fewer mutations, and those who have a slow reproductive rate. So we reproduce slowly, even though we have sexual reproduction, we reproduce slowly, which is a key factor to how fast and how likely evolution can occur in our lifetimes. Now, I know you're going to be like, wait a minute, bacteria reproduce asexually. So where are they getting this so much genetic variation from? Ah, I'm glad you asked. Let's talk about this unique, well, it's not really unique, but this key trait that bacteria are able to do. This is called conjugation. Conjugation is another word for, like, you can apply it to mammals. Conjugating is sexual intercourse, but bacteria don't reproduce sexually, but they can conjugate in the way that they gene transfer between bacteria. So I'm going to play this quick little animation so you can see.
We're going to have two bacteria. One here, this is resistant. Here's our antibiotic resistant gene. It's going to then make something called a pilus and transfer the gene over. Boom, it encodes this thing. Now we have an antibiotic resistant gene in here, and this organism now has the resistance to the antibiotics. Again and again, bacteria do this. Again and again, we see this happening in our bodies.
So this is how bacteria have survived and flourished over, the however many millions of years they've been around. They are not going to reproduce sexually, so they would have very little variation in their genetic populations. But what they do is they exchange genetic information, as you just saw, via conjugation. So if there is the chance that there's that resistant bacteria that happens to have a gene for resistance, it had a random mutation, it can actually exchange that and give that to another bacteria and spread that resistance even further. So bacteria do fall into that category of having a rapid reproductive rate, and they do have more genetic variation than you would think, being that they are asexually reproduction organisms because they have conjugation. So insects do reproduce sexually, and they have a quick reproductive rate, short life cycle. And so we've seen insects develop resistance to the chemicals that we use to try to kill them. So this is just a picture of an ugly worm-like thing called a corn borer destroying an ear of corn. This bottom picture is a picture of a person covering up really, really well because these chemicals are toxic and you don't want to breathe them in, you don't want to get them on your clothes, and he's spraying these toxic chemicals on the crops in order to try to kill the pests, the insects that are killing the crops. Roundup is a popular name of a weed killer chemical pesticide that is being used to get rid of weeds in our farms and agricultural business. So eventually it's possible that an insect develops resistance and we end up on this spiral here of, well, there's some resurgence of a pest, we use more pesticide, we are going to kill the pest, but then eventually there's going to be more outbreaks because they're going to become resistant, and then we have to use more pesticide, resistance happens, we could change to a new pesticide, eventually, that might help temporarily, but we're going to have to use more of it and more of it and more of it until eventually it doesn't work anymore, and then we see more resistance, so we need a new pesticide, and so it's just this continuous, I used the analogy of a treadmill before, a spiral is another analogy where we're constantly just trying to keep up with the resistance in developing in these populations. So here's a little visual, if you're more of a visual learner. Resistant bacteria are the red ones, regular non-resistant bacteria are the yellow ones. So in this original environment, these resistant bacteria don't have much of an advantage because there's plenty of competition for resources from the yellow non-resistant ones. But if we add antibiotics to the dish, we're changing the environment. The environment is changing, and the trait of resistance will then become favorable in that environment. So when we add antibiotics, they only kill the non-resistant ones, or most of them. So there's very few yellow left here after adding the antibiotics, but all of the resistant ones survived. So now there's a lot less competition, and you're going to see more resistant ones thrive and reproduce because they don't have competition, they have resources, and they are able to pass on that trait. So we end up changing the population to see mainly resistant bacteria when we overuse antibiotics and there happened to be a resistant bacteria. Remember that resistant bacteria just had a random mutation. Lucky for that bacteria. Not so lucky for us when we're trying to fight those infections. So they can easily survive and reproduce and make more resistant bacteria when we change their environment by adding antibiotics to that environment. This is the same exact idea, just in a different picture. So typically there's not that many resistant bacteria. Typically there's more normal bacteria. If we change the environment, and here in step 2, we add antibiotics, then we kill most of the normal bacteria and only very few normal, and the resistant ones survive. Now they have less competition and they're going to be able to reproduce more easily with less competition. and eventually see an increase in resistant strains of bacteria. And that means that they then develop a new antibiotic that can try to attack them. So these are actual pictures of Petri dishes growing bacteria. The bacteria is like this peachish kind of color in the top one. It's a yellow color in the bottom one. The little white circles are paper discs that have been dipped in antibiotics. And you can see there's a clear zone around some of the white circles. There's not much of a clear zone around this white circle here. And then down in the bottom dish, there's no clear zone around any of those white circles. So what can we interpret from these pictures? You got to be able to realize, well, knowing the facts I told you, the white discs are antibiotics, the clear regions are bacteria-free. Why doesn't this bottom plate here have any clear regions around its antibiotic discs? The bacteria are going right up to the antibiotic, meaning it's doing nothing to kill them. They are resistant to it. So the bacteria in the bottom dish are resistant to all of the antibiotics that are being demonstrated here on those little paper discs. Okay. So in this sample, the bacteria are resistant to this one because there's very, very small clear area. So the bacteria are growing right up to it. But the bacteria are being killed pretty well by some of these antibiotics. So each disc is a different antibiotic. And it represents a different drug that we've developed. So this antibiotic is much stronger and more effective on this type of bacteria than, let's say, this one down here. Okay. So the reason those bacteria-free zones are bacteria-free bigger in some places rather than another is because this bacteria does have mutations that made it resistant to some of those antibiotics. And this is why it's important for doctors to know which infection they're treating and know which antibiotic is going to be best used to target it. My dad had a really rare, weird kidney infection last year that sprang from a urinary tract infection, and he went into sepsis, which is really dangerous. It's when your body's got an overreactive immune response and it starts, kind of taking down your organs. The doctors, he had like contagious disease specialists, bacterial specialists trying to target and figure out which type of bacteria strain was causing this infection so they would know which antibiotic to use. And the first ones they used weren't working, didn't help at all, and he got worse, and then they had to change it to something else. I don't remember the names of the antibiotics, but eventually they figured out which one they needed to use to try to target it. They never figured out which infection strain it was, though. That was kind of scary, but they did get the antibiotics that he needed to work. So we keep developing new kinds of antibiotics. These are all fancy names. You don't have to know their names. We had a little bit of a gap here in terms of developing antibiotics between the 70s and the 2000s. So that time lapse was like we kind of sat on our heels a little bit too long and we didn't discover and make any new antibiotics. And we actually started seeing an increase in resistance. So you can see these graphs compared here. The decline by years in our new antibiotic development is pretty directly related to the increase in antibiotic resistance. Okay, so when there's more and more resistance, we have to develop new antibiotics. We cannot fall behind on it. So here's just another time frame of when we develop certain antibiotics timeline. There's some big gaps there in the timeline. And these resistant bacteria are really dangerous and they can cause death. There's one that's common if you've ever heard of MRSA. Not that it's common, but it's something you might have seen in the news or heard of. There's actually a brain pop on MRSA. So MRSA is a resistant staphylococcus infection. It usually affects skin, and people get like really bad skin, like lesions and stuff. And it's hard to target because our drugs don't work well on it. So how do we prevent this or slow this down? Because we can't completely prevent it. You can't stop mutation. Mutation is random and spontaneous. But what we can do is take a few key steps in using antibiotics or not using antibiotics when they're not necessary. That's one of the key steps. In fact, that is the first step. You should not take antibiotics too often. You should do your best to not get sick, first of all, and practice good hygiene, strengthen your natural immunity. You can fight off many infections. If you have a healthy immune system, you can fight off infections without antibiotic use sometimes. So first rule of thumb is just try not to take them too often. Try not to need them too often. The second rule of thumb is don't take antibiotics if they're not directed to be taken by a doctor. So if you have leftovers or a family member has leftovers and you think, oh, I'm sick, let me take these old antibiotics. No good, not a good idea. A lot of times you're sick with a virus And a virus is not going to be affected by an antibiotic. And you're just going to be kind of overusing that antibiotic and providing possible opportunity for resistant strains that happen to just be there to have an advantage by using those antibiotics and taking away their competition. Another rule of thumb is if you get prescribed antibiotics, you must take the full prescription. Don't just say, oh, I feel better now, I'm going to do a good thing and not use any more antibiotic. No, that's actually a bad thing. The doctors know how long it takes to kill an infection with a specific type of antibiotic. So if the doctor prescribes you 10 days, you take it all 10 days. I don't care if you feel better by day 7. You keep taking it. You might feel better, but you might not have killed the entire infection. And if you stop taking the antibiotics, you're going to see a possible resurgence of that infection, and then you'll need to use more antibiotics again, and we'll end up overusing antibiotics more than we needed to. Okay, giving the possibility of, if any resistant strains were present, giving them the possibility of having a survival advantage and persisting in that environment. Another thing is you don't need antibacterial soap to get rid of germs. The whole premise of soap was around way before antibacterials were. Soap is, by its biochemistry, a lipid, and it's able to pull the germs and the grease and the dirt that the germs are attached to off of your hands. So if you just use regular soap and hot water to physically remove the germs and wash them down the drain, you're doing even better than using something that's an antibacterial that kills 99.9% of bacteria. Guess what? That 0.1%, they cannot account for mutations, spontaneous mutations that are unstoppable and spontaneous and random, and we cannot account for them. So you will never find a product that claims it kills 100% of pathogens because we cannot account for mutations. So regular soap physically removes and pulls off that material and is therefore better to use because therefore we're not overusing antibacterial products. And the last thing you could do is you can shop for food that has been made by animals that were not given antibiotics in their feed. So the idea of being a farmer and a rancher and you grow pigs and you grow cows and you want to keep them healthy, the ideas where farmers would say, all right, let me just give antibiotics to my pigs and my cows so that I can kind of ensure they don't get sick. Keeps them healthy, keeps my margin high, keeps me getting money. But what you're doing there is overusing antibiotics when they're not needed. And you're only killing non-resistant bacteria. If there were a random spontaneous lucky bacteria that happened to survive those antibiotics, you're giving it the survival advantage by taking away its competition. So we know, and we see from a health perspective, it's best to not do that. It's best to not give antibiotics to your chickens and to your cows and to your pigs, not just for your own benefit, but for the benefit of the functioning of antibiotics in general, for the world in general. So this is a little grading scale of which companies, this might be a little bit out of date actually, I probably should double check and see if I should find it. I found this thing like 3 years ago maybe, so I don't want to put down everybody in the F list. Maybe some of them have stepped up and increased their suppliers and are using different suppliers, but if this is something that's important to you, it's something that you can consider. I know those who advertise it, Panera and Chipotle, they put it out there, they make it a big deal that they don't have any products, any meats that were given antibiotics in their feed. They're very big on organic, no antibiotics. And that helps just health in general, the world in general, to help prevent antibiotic resistance. And that's the end of my story. So if you have a question, you are welcome to unmute yourself. And I am going to end the recording now.